Hi, thanks everyone. Thanks for letting me speak also at SOTA, UAMing. Um, some of y'all probably recognize me. I'm a former co-chair of SOTA and also a member of the board of directors for MANA. My name is Daniel Hitchcock, and I'm a software engineer at the Broad Institute, or at the Metabolomics platform at the Broad Institute, directed by Dr. Clary Klisch. And today I'm excited to talk about something we've been working on for quite a while and something we've started using internally almost exclusively now for part of our processing pipeline, our new targeted feature extractor. So today, I'm just going to go over just real quick what feature extraction is. I think most people know what it is, why we need to do targeted extraction, why we developed our own tool to do this, give a brief demonstration of it, and then go over some of the results that we have. So just an overview of this is kind of how I view processing metabolomics data. The first part's acquisition, where you have the raw instrument data, then you have feature extraction, which goes from raw files to a feature table. Then you can annotate the data by a variety of means to get an annotated feature table. And then lots of additional processing can happen afterwards, including QC, drift correction, filtering, clustering, and obviously analysis too, if you actually want some results or findings from your data. There's also a lot of language that goes around. So this is kind of the feature extraction. Also, people can call it peak picking, peak integration, processing, pre-processing. Just a lot of synonyms, and I might use them interchangeably during the talk. So non-target extraction is usually the go-to for experiments with Orbitrap and TOF data or anything that's run in full scan mode. It's great because you can extract thousands to tens of thousands of features and do this in an automated way. So you can have a feature table. Usually it won't give you, you should just describe the metrics that are there and just produce both knowns and unknowns. I have leucine and isoleucine shown there as an example. The other great thing about it is that it enables the discovery of new metabolites. So aside from the scale, you get things that are measured that we don't know the ID of. Shown here on the right was one study that our group was a part of, looking for diabetes biomarkers. The blues are the reds are the known features or targeted features, and the blues are the unknowns. And one of which was a signal that was highly associated with diabetes that in our group was the structure was elucidated by Julian, our associate director. And it was just a cool case of finding a novel metabolite, through non-targeted data. We wouldn't have found this on a triple quad because we weren't looking for it. There's experiments and everything. But one of the problems, though, non-targeted extraction isn't always perfect. And people have gone into great detail probably about some of the things that can happen. I kind of put these in three categories. The first is what I call dropouts. And we use this lingo internally, and maybe other people do too. Shown here is a sample profile plot of about 300 samples from this IHMP2 data. Oh, and I put 2018. I'm not sure if that's actually the date on it. It might be 2016. This is the data that's in Metabolomics Workbench, though. And it's a rough chromatography. It's HILIC-neg run on a Luna column, mostly for looking at central metabolites or a citric acid cycle. In this case, here is a SPAR gene, which is the left peak. And it reports it as, oh, and sorry, these are the samples that don't have dots. And then the blue and red dots are the pooled references that we run every 20 samples as QC. So they're just technical replicates from the entire study pool. This one would show that the SPAR gene is 0, but clearly there's a peak there for that sample. Meanwhile, this one's even smaller, but it is reported. And then these are just total dropouts too. Another thing, this is kind of similar, maybe not the best example, but misintegrations. This shows these are present. It's hard to say if those are accurate, and certainly this one is a dropout, and those two, they should all be the same. And, hard to say what's going on there, but it looks like it might have been misintegrated for a variety of reasons too. It's a challenging issue. And sometimes things are just entirely missed. There's benzoate, which is, I guess, in a lot of plants. This is the raw data, but the non-targeted data set processed through QI, Progenesis QI, which is what we use. Salicylic acid, which is, I think, a downstream metabolite of aspirin. Hippuric acid, a marker of gut health. And aminoadipate, which is a biomarker for diabetes. So When these features get missed, if they need to be retrieved, you'll generally want to use targeted extraction. So that brings me to this point. Reasons you might want to do that is to rescue or inspect a feature. So this was, I forgot which feature, hippuric acid, and that's what the sample profile looks like if it's pulled out manually. There's a lot of vendor-specific software that work with their own instruments that's designed to do this, and they all are great pieces of software too for exploring metadata of the acquisition. Next, you might want to process triple quad data, which is kind of the standard workflow in my understanding. I'm sure there's non-targeted ways, but most of the time I've seen, using manual peak picking and guided approaches. There's also like stable isotope labeling, which might have very low abundance or messy peaks that need to be pulled out. I'm just kind of like just going over some general ideas. And finally, you might want to inspect some public data. In this case, I doubt anyone has licenses for all these software packages, so they'd have to turn to open source packages to try to view these. And there's a couple that are out there. I've actually never heard of Open Chrome before I started looking for more packages, and something I need to look for more. We had very specific needs, though. For one thing, we run a lot of population studies. I think our largest one now is comprised of multiple cohorts and something like 13 or 15,000 samples. But dietary biomarkers, TopMed, and MotorPack are some of the larger cohorts or studies that programs we're a part of. And we usually run these in batches and columns. We align them with Eclipse, which software we released this year. But problem is, if we miss a feature in one batch, then it kind of misses it in all of them. And we have to go back and figure out how to retrieve that feature. So this, if there are random dropouts from the non-targeted data, then if that randomly happens to a feature, we have to go rescue it. And there's other ones too that we know are difficult chromatography. So we needed scale for this. And just kind of a quick table over specific needs and how it compares to proprietary and open source software packages. And not trying to like call anyone out or say we're better. These packages all have great uses. It really doesn't design for like large scale processing though. We need to be performant. Tracefinder is usable, and we used it for a long time, and it's great, but it is a bit slow. Open source, I haven't found a package yet that works with more than 500 samples or so. We needed click and drag integration, like, be able to quickly swipe where we want those. I think the open source ones that I've seen are limited just to like integration ranges, which is a powerful way to integrate peaks. We also need to be bare bones and simple. We didn't need a lot of features. And I have this at the bottom. We don't need annotation, MS2, drip correction. We really have those solved somewhere else. So we wanted this to be very basic, very fast, very lightweight to get up and started. And we also wanted to be able to handle other files too, triple quad, TOF, and thermo raw files, and mzML files too. So with that, I'd like to introduce a screenshot of our software. We haven't come up with a great name yet, but this is TFE. If you've used Tracefinder or Multiquant, this would probably look a little bit familiar to you. In one panel, this is the main view that we use to look at the data and inspect it. We have our annotations here that we provided. We have a list of the samples and metrics on how the peak was integrated. And then we also have a window of every XIC or extracted ion chromatogram that's there. And two other quick screenshots before I go to the demo. There's also a samples page. That's just other parts of the program, and the annotations. We do have a peak integration algorithm that we came up with that could be better, but it works. And I'll go over that more later. And it also uses like pop-up windows to be able to modify in bulk everything that you're looking at. So some of the features are it's cross-platform. We wrote this in Vue, so it's basically a web browser is what you'll be looking at in Electron, and fast parts are written in C. We've tested it with, I think, like 8,000 samples so far. Even at 5,000 samples and 500 annotations, it only takes 20 gigabytes of RAM to run. We can also take Orbitrap and triple quad data, and in theory, we can take mzML files for TOF, though I learned we need to work on it. It's kind of funny what happened, but each scan is its own scan filter, basically. We need to be shareable, able to report this stuff and be able to share data without sharing this, gigabytes of raw file. easy to use, maintainable. One thing, we use this now at the Broad, so it's not a project we're about to abandon anytime soon. And then we also needed raw and mz support. We collect a lot of raw files, and it does take up a lot of space and a lot of time to convert files to mzML to be used. So, um, just a quick thing, we do separate the idea of extraction versus peak integration, and we cache things in memory to make it a lot faster. We only pull from raw files when needed, and there is a semi-automated integration algorithm that can be overridden very easily. And a couple of things before we get to version 1.0 that I'll put this slide later just to let you know well, it was supposed to be later, but before we get to the demo, we need to cross-compile it and test it on Linux and Mac. We have a bug to fix with TOF files or mzML file, as TOF files on this specifically. Updates to the algorithm, display issues, licensing, and this is kind of important too. We need like maybe a little bit of guidelines or instructions because if this gets unleashed into the world, I don't know, people have creative ways of using software and integrating peaks. So demonstrations I'll show a very small example with some plasma samples from various studies, just our pooled references. These will all look different with 879 annotations. I'll show stuff from the IHMP2. I know I said there'd be 500 samples. I forgot it was processed in 2 batches, so we process it 1 batch at a time, so it's only 300. And then a stress test to show that it's capable of working with 2,000 samples and 500 annotations. I'm not saying that people would go in there and do that many. Like, I think that would be insane, but it is possible in theory. So I'm going to lower the screen and start an example. So the first example is just these raw files right here and an annotations CSV. And these are peak picking settings that are kind of default. As you go over and learn the chromatography over time, you can fine tune them and reuse them. But here I have 880 annotations. So, okay, I'm going to open that directory. Short, okay, great. The first thing it'll do is find the raw files that are present and then and show them on this page. Here's the annotations that are present too. And I'll click extract. It'll go through and pull XICs for everything. So all 800 are pulled. And then this is the actual integration algorithm. And then if you click, you can see the peak table right here. So a couple things you'll notice. This looks okay. You can see that there's a slight kind of tail right there, and maybe you want to grab that or not. If you wanted, you could just click and grab that over and just drag it. Or let's say you don't want to do that. You actually want to open a pop-up window and maybe, I don't know, maybe, let me see. Oh yeah, smoothing. So if you just kind of increase the smoothing, it's stuck on the local minimum right there. So you could increase the smoothing. This uses a Gaussian kernel, and it's okay to smooth data as long as they're in equal scan lengths. Apply to all. Oh, whoops, I have my annotations thing open and I got mad because it updates that list. There we go. Okay. And there we have this peak. Next, I want to go to pantothenic acid. Okay. So this is a much messier peak, and this again is a difficult region of chromatography on a difficult chromatographic method. So it's very jagged. What we could do in this case is say we wanted to do just like a range integration. Yeah, I don't know, just increase the range. Up to you. Or let's say we don't like that. We can increase the full width half max, which is kind of part of how the peak algorithm works. Increase the baseline search window. Still not good enough, so then we'll have to maybe smooth it a little bit too. And I haven't found an argument against smoothing it. If you know that it's the feature, we're not destroying signal, we're not inventing signal, and these integrated values end up being about the same. So hit apply, and everything's integrated. I'll just drag that one over. Okay, great. Next one, EE. E dash 38, 4.
Sorry, Daniel, while you're pulling this up, when you're changing a parameter like this, in this case, the smoothing, that it applies it to all of them, to all the study files. Is that right?
Yes, that's correct.
Okay.
And yeah, and things should generally be smooth the same. If they are the same scan length, it'll be all right, but you don't want to have one that's smooth and one that's not. And
Yeah.
it is
Yeah.
possible to do that though with an apply to one. So here we'd want to increase the range, maybe peak search range, and I mean, I don't know, that works. And it grabs the peak, you'll see a tail missing there. And I'm building to something. I'm not just going through a bunch of boring integrations at this point. Here's another challenging one. And these are all run on different instruments again, so this is a much later peak. You could increase the range. You could do an apply to one. Let's increase this range a little bit. See these red lines out here? That's the range that something needs to be. And if you exceed that, it gets mad and says you need to increase your extraction parameters. So I'm just going to increase that a little bit. Okay, cool. Apply to all. And now it doesn't do it automatically because I don't want to lock people into a long process if there are 1,000 samples or thousands. And maybe I'll make this an option to automatically do it. But let's go ahead and hit re-extract, reintegrate, and there's this. So these were run at different times. You might have observed that some run early or late, and some run early. There's a very experimental thing I have in here for retention time scaling, where I can take those peaks that I just determined, PE 38. Yeah, there might be a better way to implement this. Oh, that one's missing, isn't it? Why? Well, whatever. And N6, methyl lysine. Okay. So I don't know why these are missing, but I might have just grabbed the wrong one. Oh, I did. Sorry. P-38. PCP. Okay, cool. I add that. I'm gonna drop this one and then add back in 6 because I never actually did add it. Okay, save and apply. And now everything needs to be re-extracted, reintegrated, but we end up with a bunch of nice scale peaks that all match the same retention time. This only works if your chromatography allows it and you choose the correct, and you know how the scalars work and you don't choose a peak that has a weird behavior. Okay, so that's the end of that first example. Next thing I wanted to show was how easy it is to share the data, at least in a perfect world where everyone inspects everyone's work. Okay. Oh, and I forgot. Here's how you get results. You choose which file you want your reference to be, which you don't even have to. I'll just choose one and then click save, and it will save. Well, it's supposed to save it. Oh, I had the wrong directory, don't I? Oh, sorry. Let's save. There we go. Export. Okay, there we go. Sorry, I forgot how to use my own software. All right, the next example. So here's an XIC's bin. This is actually for the data I was talking about a minute ago. And all we have to do, let's say I wanted to see the peaks that were integrated, and I got sent this xics.bin. I would just open this directory, and it will create the sample. It will infer the annotations from the bin file, and tell you everything's missing, but it will allow you to go in, inspect everything, and even change things if you want. Yeah, so you can change it. What you can't do though is change the extraction parameters because we don't have the raw files. But it's great for just being able to show where these integrated peaks came from. And I was just going to show with this one just kind of what the chromatography looks like. This is our more difficult method. The other one I was showing was actually our easier, our smoother looking method. But Oh, here's 2-amino adipate, by the way. And Carrie was one that integrated this, and she did a lot of things like, manually clearing things out because we didn't think that was the right peak. And and also you can do things like, let's say you want to remove everything below this amount. You can do that, which will now clear anything below a value, since you can sort by area RT, or you can just revert everything back to what the program wants. And so this will bring back what your library annotations are. And if you have a manual annotation and you change something somewhere else, well, this is a bad example with the smoothing, the thing I said not to do. Let's say it's that. It won't override the manual smoothing. Or sorry, the manual integrated value. Okay. The final thing I wanted to show with this demonstration was just a stress test. Here is 2,000 files from 3 different studies on looking at dietary biomarkers in plasma or finding those from people fed controlled diets. So this is isoleucine. I already pulled it up because time is limited, but here's me scrolling through 2,000 things that are viewed. I can adjust one if I wanted. Slightly laggy, but totally usable. I can click on it and make adjustments, explore. I can also sort it pretty quickly. So here is everything that had low abundances, which actually kind of reveal the problematic peaks. And if I wanted, I could update it and apply it to all. It does take a little bit of time to do that. In fact, I'll show what it takes to switch a metabolite. I need to figure out how to make this quicker, but right now it's fetching extracted ion chromatograms from the back end. That's what that says. It might be something to do with like FFI overhead or the form function. Yeah, reading the C code, basically taking too long. And it will take 30 seconds, which seems like an eternity when I'm giving a talk. It's only 25 minutes. But in a second, it's going to show that it's drawing the items and then have them displayed. And what I'm going to do here is make a change to one of the extraction parameters once this is loaded, and just show like how useful it is that it doesn't go in and re-extract everything immediately. It actually tracks the state. So let's say I have this and I decide that my ppm window is off. And maybe I do want more range because I want to see other things there. I'll hit apply to all. It again is going to take about 30 seconds to redraw everything, and it shouldn't be integrating. But it will only take 30 seconds as opposed to 10 minutes, which is what it'll take to actually re-extract everything. And this is 2,400 megabyte raw files run on a C18 positive method. So, and that'll end the demonstration unless we just want to watch it be slow. But my point is, anyways, switching metabolites does take a little bit of time, and committing to these things like changing it. But in real time, you can make the adjustments, and then whenever you need to switch metabolites, you can stretch or get coffee or something. And if you need to extract, then you can do that also. So maybe I'll get back to the presentation. Okay. Anyways, now strong plots. Okay, right. They're all red now, and meaning they need to be re-extracted. And if I go here and click re-extract, it'll go through. This is a long process, obviously. And the last thing I'll say, our raw file reader is one we wrote custom, and it actually is available as a Python package. It's written in C. It reads the whole thing into memory and parses it into a common format. Works with mzMLs too. We'll figure out how to make it faster someday. But anyways, okay, while that's going, back to the presentation. Okay, so finally the results. Remember those peaks I showed earlier, or the misintegrations and dropouts? Here's what they look like now. Or this is the TFE data extracted, plotted against our non-targeted data. So this isn't a great correlation, and it's hard to tell what's going on. I also plotted these on log scale, and this shows that these were dropouts that for some reason our non-targeted software, Progenesis QI, didn't pick them up. Um, Again, not to call out that software, it actually works great. And there's a lot of others too. These are bad integrations, dropouts. Here's what the rescued features look like, the ones that were missing. They look pretty good. And they weren't all bad. Here's hydroxybenzaldehyde, which gave a near perfect correlation, both in raw, or sorry, say raw. This is log scale on the right. Raw means not trip corrected. And now this complicated plot. So you're probably wondering how it did with all the features that we had in this IHMP2 data. If I could direct your attention to the bottom plot, this is a list of annotations that we interrogated in TFE, and on the y-axis is its log correlation, Pearson correlation from the non-targeted to TFE data. So gabapentin, for example, is shown here, and it looks great. And actually, I ignored this side because these were unreliable peaks and they were marked out in TFE. So I didn't include them for the analysis. That's why, yeah, there's a red line there. But these look perfect. This looks okay. This looks also okay, Fucose. Going over here, we start to see a little bit of disparity at about 0.9. And then 0.8, it looks kind of messy. And these are the ones that weren't found at all, and they were set to the worst one, which was diaminopimolate. So don't look too much in these numbers because they look a little scary, actually, that this would suggest that only 58% of the features are accurately measured if you use whatever arbitrary cutoff. But this is not an accurate evaluation. It's just one program, one sample set on a very hard chromatographic method. It's also just one metric. Like there might be better options than like Pearson correlation. Also, our list might be biased to QI. These are things that we had trouble with historically, so they populate our list, although there are a lot of normal features that do great in either. Also, you might not agree with how we integrated those peaks, but at least it's transparent, and you could prove me wrong if I showed you the data, which is the whole point of TFE. So um, the software was developed in our group by our engineering team, myself included, and then Jesse, Craigie, Chloe Sturgeon, who is pursuing more computer controls careers now outside of biology, and Giant, who worked with us as a high school student. He's in his second year at Brown now. Carrie did a lot of the work with integrating and helping generate that plot I just showed. And then our directors, Clary Klish is the platform director, and Julian, who worked on the structural elucidation, our associate director. But thanks, everyone. That's it.
So thanks for having me. I noticed some familiar names in the chat or in the participant list and some unfamiliar. My name is Corey Broeckling. I work at Colorado State University. I do run a core metabolomics facility. We do proteomics and ICP-MS as well. But I spend most of my time thinking about metabolomics, and this is a description not of software, but a data resource which is designed to enable evaluation of software. So I'll start by thanking all of my collaborators. There's some members of my group here on the author list, Linsing and Nathan, but in addition, we have members from Gary Patty's lab and Jushia Du's lab. And this was a collaborative effort that it's been a long time in developing, quite honestly, which originated out of the NIH Metabolomics Common Fund Consortium and some of the supplemental funding that came with the grant that we were on together, where we wanted to put together a resource that will enable benchmarking of metabolomics software. So that's the quick preface for what we're talking about today. I think you're all familiar with this. Metabolomics data is complex, messy, but what I want to do is delineate why and specifically outline some of the problems. So when we're going through metabolomics processing, we have the first step is typically centroiding. Sometimes this happens on the instrument. Sometimes it happens post-data acquisition. We have to detect features, determine which features line up with each other, as I think Daniel was describing in the last presentation as well, correct for things like retention time drift and quantitative drift in instrument. We also sometimes want to be actually grouping features together. So we have multiple features from one compound. We want them together. We want to annotate the ion. We want to annotate the compound. And we would love to be able to evaluate quantitative precision and accuracy to be able to benchmark things like integration imperfections more reliably. So benchmarking as a process is really the process of given raw data and ground truth, you evaluate the performance of your software. What are the processing steps and how well did they convert raw data to ground truth? What was that process like, and how accurate are the results? And your evaluation is some sort of combination of true and false positives and negatives, where your true positives and false negatives are defined by your ground truth and how well you recover that. This is really hard in metabolomics. And part of it is the complexity that I described, but specifically, there's some really challenging areas of metabolomics that make this difficult. One is that the small molecule composition of any really complex sample is incompletely known. If we're given a human serum sample, whether that's a NIST 1950 or experimental sample, we really don't know the ground truth as to what small molecules are present. So big problem number one. Contamination is a big problem. A lot of contaminants are actually small molecules. And so we can be saying, well, even if I know the ground truth of what's in 1950, do I know the ground truth of what I've introduced in handling 1950? And the answer is there as well, no. And just as importantly, the contaminants are often small molecules. In fact, some of the contaminants from plasticware are actually biological compounds as well, metabolites. Another problem that we have is that many mzML libraries, authentic standard libraries, are too small. And at the same time, PubChem, a structure library, is too big. And so we don't have a great ground truth reference set that we can be guiding annotation performance by. We also have these paired problems. So we have a one-to-many problem where one metabolite can generate many signals. And we have a many-to-one problem where many metabolites can look like the same thing. A lack of resolution chromatographically or mass spectrometrically can result in one signal for multiple compounds. And finally, one of the big problems where proteomics really has it good is that the proteome maps to the genome. And that is much less easy to do with confidence, especially in metabolomics as compared to proteomics. And of course, we have the fact that we're dealing with mass spectrometry. There's a good deal of noise, so we have to be able to deal with that. So the goal of our process was to try to generate benchmark sample with all of these caveats in mind. And so a benchmark sample set, a benchmark data set, where the approach that we took was that the structure of the raw data helps us to define a ground truth in the face of all of these challenges. And I will describe what that means. So I like this term, structured complexity, where we acknowledge that metabolomics is messy and that there are a lot of signals that are difficult to predict and define well. But what we can do is we can take advantage of that complexity and build structure into our sample set to enable us to help establish ground truths. And so the way that we're doing this is we're taking real complex samples. And in this case, we're using SRMs. 1950, 8231 is one of the more recently released experimental at the time SRMs, where we have two different standard reference plasmas that we're using. We also have yeast extract, which we purchased from Cambridge Isotope Labs, where we have a C12 and a C13 version. This was to help support a credentialing type approach that Gary's group has developed. And we have authentic spiked samples. And so we actually commercially bought 96 pure standards from a company called Chromadex primarily, but some other sources as well. And we actually generated two relatively complex standard mixtures. We'll call them Spike A and Spike B, where they all have the same 96 compounds, but the concentrations of each of the compounds differs in A versus B. And so you can imagine it as sort of a synthetic complex mixture of two different types, much like we have paired 1950 and 8231. We have paired C12 and C13 yeast. We have paired 96 compound mixtures. We also built into this data set with a lot of feedback from the metabolomics group that we were working with, groups, many labs, probably some of them on the call here, the incorporation of internal standards, isotopically labeled. We have a pooled QC sample that we're utilizing in all of our data acquisition. We have blanks, and we'll use this concept of structured dilution series, as I'll show you in just a second. So this ultimately is how we structured the sample set. And the way that this figure outlines is, if you look at the bottom here, you can see every column is a reagent. And in most cases, this is a complex reagent. So we have SRM and 1950 and 8231 on the left. And can you see my cursor? Yes?
Yes.
Okay. Thank you. So the cursor, I'm just going to highlight this area here. I'll start at the top. This is a dilution series of 1950. So what this represents is we have 4 different vials, 4 different samples, where we have a dilution series. In this case, it's a 50-50 mixture of 1950 and 8231. And then we dilute that several-fold. And what that means is, even if we don't know what is in 1950 and 8231, we now have an expected signal intensity trend that we can look for when we look at the samples in this dilution series. That is, at decreasing concentrations, we should have decreasing signal. So all of our real signals should show that trend from 1950 or 8231. That is a dilution series. Now we also have a reciprocal dilution series, which I'll highlight here. And in this case, the total SRM 1950 plus 8231 is constant in microliters of volume per sample, but the proportions change. And so as 1950 decreases, 8231 increases. And so that gives us another structure to look for signal intensity changes in these samples, even if we don't know what the compounds are. We did this with all of the reagent pairs. So you'll see dilution series for the yeast, C12, C13. You'll see dilution series for the 96 mix. But you will also see blocks where now in this case, the samples, the SRM 1950, 8231 are both constant at 50% each. But then we add in the spike A, spike B 96 standards mixture. So now we have spike A, spike B in a reciprocal dilution series here. We have the exact same 96 mix A and B in a reciprocal series here in the presence of 1950 and 8231. So it enables us to really test the impact of complexity on our ability to process these data. And we also have process blanks built in here, just pure solvent blanks. So process blanks are taken through sort of an extraction evaporation process, and then solvent blanks are just a clean solvent. We also have QC samples, which are equal portions of all of those reagents. And we have extra QC samples so that people that are using the QC samples for example, for normalization, drift correction, et cetera, you can actually have test QC samples by which you can evaluate your processing that were independent and not used in the normalization process. So it becomes a relatively complex sample set, where in this case, I think that we were about at around 50 samples total, something along those lines. I don't remember the exact number. But we also wanted to replicate this. That doesn't replicate a real experiment. And so a real experiment, we might have 3 reps of everything. And so we have now every one of these samples is in batch 1, in batch 2, in batch 3. These were run back to back to back. And then to enable things like, if I have more retention drift, can I deal with that? We have a batch 4, which was acquired at a later time point. So we ran these 3 sequentially, waited, I think, in our platform for LC-TOF about 6 weeks, and then we acquired batch 4. Same sample set, just space between them to help push the limits of things like retention correction. And then things get really big because we wanted to do this on different platforms. And so people use LC Orbitraps, people use LC time of flights, people use GCMS. We ran it on all of them. And we ran it in the LC methods in positive and negative mode and in reverse phase and HILIC. And so what we've generated then is a sample set which is based on commercially available reagents with a recipe that anybody could make if they ever wanted to do so again. So we wanted to make sure that this was reproducible in the sense that all the reagents are available. And secondly, is that we wanted to make sure that the data is representative of the types of data that people are actually analyzing. So we couldn't just use reverse phase LC because there's a lot of GCMS metabolomics out there as well. And we wanted to make sure people had a data set for that. And so we ran it on all of these platforms. So just a few notes on the data. All the run order is randomized except for the pooled QC samples within each batch. We acquired MS1 only data on those LCMS platforms initially at 3 hertz. So we made sure that we got 3 MSMS per second on average to make sure that we had decent chromatographic coverage. All the data is acquired in profile mode to enable people to look at their centroiding algorithms. In addition, we acquired data-dependent MSMS on every one of the 6 reagent ingredients that I described earlier using iterative precursor exclusion. And so this will help to enable annotation efforts using standard MSMS type approaches and to help establish as close as we can get to ground truth on annotations. So Zhujiadu has been handling all of this data. All of the data are generated from LC TOF, LC Orbitrap, and GC for all of those methods. And Zhuzha has been working diligently on getting these data onto Workbench. And so the idea is that all of these data are available to anybody, anytime, anywhere, for any reason. And the GCMS data is up there currently. I think the Orbitrap data is uploaded, but the package isn't quite available. And the LC-TOF data, I think, are currently being uploaded. So we're really close to having all of the data released in its raw form. The data package is outlined here. And so we have all of the raw data, which had to get zipped and delivered. These are the directories that the data is structured by. This includes documentation, including the design spreadsheet, which is a graph or an Excel spreadsheet version of that figure that I showed you to show you the composition of every ingredient in every sample. How much is there? All of the detailed methods are there. We have a README for guiding you through all of this. We, in addition, have that Excel metadata composition in this document here. Raw files in the native vendor raw file format. So that's a Waters raw for our QTOF, a thermo raw for GC, and thermo raw for Orbitrap data. But we also have MZ profile, so not centroided, and then centroided using the vendor software. And so we have each of those 3 forms of the raw data available. Now, one of the challenges that we're dealing with is we want to make sure that this data is of high quality. And the question that you might ask is, how do you make sure that you have high quality data when you have to process your data to determine the quality? And we're trying to make this available for benchmarking processing. And it's challenging. So any gold standard that we assign as far as a benchmark is going to be partial at best. So we acknowledge that there's still challenges with benchmarking. This is not the be all and end all, but it's a nice resource. And I'll show you some examples as to how we can use this. And we want to describe an approach toward developing that gold standard. And so all of these steps that I outlined before, in theory, we should be able to evaluate using the data in hand. And I think that's the important part. The reason it got so big is because there's a lot of steps that go into metabolomics processing. And to evaluate them all, you really have to build a relatively large data set to enable that. And so the data becomes large, it becomes a little bit unwieldy, but there are subsets of data that you can use to evaluate some of these. So our first step was to evaluate data quality. We want to keep the data processing minimal. And to say, is our data of high quality? Does it reflect what we put in? And so in order to thinking through how to do this, this is one way to approach it. It's not necessarily the only way or even the best way. But let's just make the assumption that the total detected signal should be dependent on the sample composition. And so then that batch and run order can also impact those quantitative values. And so what I did was I processed all of these for the LC-TOF data. using XCMS, using the vendor centroided mzML files. And I use XCMS heavily, so that was sort of my native platform for doing this. And so we can actually just use an ANOVA model for this. And we're saying the total XCMS signal should be dependent on the amount of 1950, the amount of 8231, and all of the other ingredients. And to say how often does that ANOVA return a nice significant result? And the answer is, in most cases, the answer is yes. And so every one of these here is a p-value for TOF, reverse phase platform, or HILIC platform, positive or negative mode. Each factor in this ANOVA will have a p-value. And you can see that there are all of the ones in bold and italics are significant. All of the ones that are not bolded are not significant. The ones that are not bolded are marginal. So we're at about 0.08 here, and this is the highest one at 0.14. So these are the spike compounds, which are the simplest reagents. So we're borderline on just one or two platforms on the negative mode data for those, where we have fewer compounds and fewer ions forming. Because we're in negative mode, it's more selective than positive mode generally. And so this gives us a sort of a big picture, crude estimate of data quality, that the total signal from all of these chromatograms seems to reflect the experimental design really well. There's another way that we can look at this now. So we can take all of these data and try to look at, for example, the impact of sample complexity on our processing. So if I take it through a workflow and I take advantage of those patterns that I just showed, where we have our 96 mix both in a reciprocal dilution series clean and in the presence of SRM 1950 and the other SRM, but also in the presence of the yeast. And so we can look at the impact of increasing complexity on our processing efforts. And so if you look at the PCA for our reciprocal dilution series, you can see nice separation. I apologize for the small font, but we're going from the spike A concentration of 0 on the left to 1 on the right, 100% on the right. And we see a nice trend left to right on PC1, which is exactly what we hope to see if everything's working well. Now, if we look at that same reciprocal dilution series in the presence of a plasma background, things get more messy. And so you can see now the 0 is on the top. We're going down as we increase the concentration until we see the pinkish purple on the bottom, sort of bottom right here. So now we're getting separation on PC2, and there's a little bit of an overlap here between the 100% and the 75%. So that's the impact now of the plasma background. In a complex background, it gets more difficult to pull out these signals. If everything was going perfectly, that probably shouldn't be the case. And we put it in a more complex background still in the presence of yeast, and things get messier still. So this is just an idea of how we can use these data to really try to evaluate our workflow. And what this is telling me is that my workflow for pulling out the sensitivity for these particular signals is being impacted pretty heavily by the complexity of the background. Now we can also do what I did for that initial total signal and look at the individual compounds. So here's an example, just for one compound. I'm going through a process here where I'm taking XCMS data. I'm using RAM cluster to cluster those features into compound spectra. And so compound 5 is just the 5th compound in the data set. As arbitrary, we haven't assigned it an annotation. It has a spectrum, MS1 spectrum, and signal intensities for all the samples. And we could use that same model that we had used before. And now that what we're looking here at, showing here in the plots are effects plots, so the fit regression for each of these, for each factor. So what we can see is that for this compound 5, there's very low effect of SRM 1950, a slight slope, huge error bars or uncertainty in that linear regression fit. Same for 8231, yeast C12, maybe a slightly better fit, and yeast 13C maybe a little bit worse. But we see a nice clean signal for spike A and spike B. And if you look at the p-values, these are highly significant, and these are not. And so what this is telling us is, compound by compound, is that this compound likely represents one of the compounds in our 96 mix spike samples. And so you could do this for every compound now. And you can make the assertion now that every compound should be linear dependent on one of the reagents at least, if it's coming from one of these reagents. And so you can now look at for all of the compounds, and in this case, we have for the initial processing, over 6,000 compounds were detected after the processing. Approximately 5,000 of them were linear. So we're at about 76% were linear. I then took it through some processing steps to say if we did blank filtering, to say we have to have, it has to be present in one of the samples at least 3 times higher in the QC samples than in the blank samples. And if we eliminate all those that are not, we've eliminated approximately 3,000 compounds using that filter. And maybe that's too much, but that's the result of it. And if we eliminate those that are removed by blank and look at the linear, now our 95% of the compounds are linear. And so we can see that by doing that blank filtering, we've now cleaned it up so that we've got a lot of compounds that are detected still, over 3,000, that are mostly quantitatively similar in the blank. And the vast majority of them are linear with respect to at least one of the ingredients. We can also go through a QC normalization process where we correct for things like instrumental drift in signal intensity that improves our linearity a little bit. And we can go through QC filtering to say, I need reproducibility in my test QC samples with CVs less than, say, 30%. And now all of a sudden, we're increasing our linear responses up to about 99% of all the compounds that are still in the data set at this point are still linear. So what this seems to suggest is that these filtering steps are removing the poorly performing compounds, which is exactly what they're designed to do. The downside is that while we're increasing the percentage of compounds that are linear, so we're up to 99% here, we also actually have a pretty high false negative impact. So we started out with almost 5,000 linear compounds, and we ended up with under 1,700. So we've thrown away about 70% of our linear responding compounds. And so this also highlights one of the challenges with coming up with any gold standard benchmark by which to compare. Right? So if you're evaluating your processing software, the question is, which of these is the right answer? And I'm not sure that I know the answer to that. But we have a data set and we have a process which will enable us to evaluate processing performance and the impacts of it. In the interest of time, I'm going to skip through this. All this is showing is that this holds for multiple data sets. So I was showing reverse space positive. Those trends are true for all of the TOF data. And actually looking at not only the p-value, but also the quantitative trends that we're looking at, where the more processing steps you add, the higher percentage, but the potential burden of false negatives is quite high. So I'm going to blow through that. Future work. We got a lot to do still. The data is almost at Workbench. I know Juju is close on that. We want to get that data out with a full description with a target journal of scientific data, which is a format designed to highlight data resources. Our goal with this paper is not to show you the best way or our way to process data. It's to highlight the resource itself. I've got all of that work that I did on the TOF. We got to do that on the Orbi. We got to do that on the GC. This is for some of the R-based work. Everybody will have their own platform by which to evaluate this, and that's what it's designed to be. We're putting it out there in a format that any software should be able to take in. We also have to do a little bit of curation on the 96 mixture collection for signals and the internal standards. Those are ongoing, but we're pretty close on that, and that's something we definitely want to put in the target journal as we write it out. With that, I think I will stop. I want to acknowledge NIH funding for this. So this, as I mentioned, came out of the Metabolomics common fund work on a grant that Zhuzha and I were on, and we wrote a supplement to it with Gary, Patty to help purchase reagents, get all the data acquired, and get the data out there. So gratitude to them for that. And with that, I'd be happy to take questions.